Mitosis is the process of cell division of eukaryotic somatic cells, which is preceded by interphase and divided into 5 stages. Interphase is separated into 3 phases, G1, S, and G2. These phases account for 90% of the cell cycle. During G1 phase, the cell increases in size and the organelles start to duplicate except for the chromosomes. In S phase, the cell continues to grow and the chromosomes are duplicated. During G2 phase, the cell continues to grow in preparation for mitosis. At G2 of interphase, individual chromosomes are not observed and 2 centrosomes have formed. A nuclear envelope bounds a nucleus which contains 1 or more nucleoli. Prophase occurs when the chromatin fibers condense into discrete chromosomes. Each duplicated chromosome appears as 2 identical sister chromatids joined together at their centromeres. The nucleoli have disappeared. The mitotic spindle, composed of centrosomes and microtubules, begins to form as the centrosomes move away from each other. By metaphase, the nuclear envelope has fragmented. During metaphase, the centrosomes are at opposite poles of the cell, and the chromosomes are lined across an imaginary plate known as the metaphase plate. Each sister chromatid is attached to kinetochore microtubules coming from opposite poles. Anaphase begins when the 2 sister chromatids split and each become a chromosome. The daughter chromosomes are pulled to opposite poles. By the end, at each pole, there is a complete collection of chromosomes. During telophase, 2 daughter nuclei form in the cell. The nuclear envelope begins to form and the nucleoli reappear. The chromosomes become less condensed. Mitosis is now complete. Cytokinesis occurs when the cytoplasm divides, causing 2 diploid, 2N daughter cells, each genetically identical to the parent cell.
Meiosis is the process of sex cell division, and like mitosis, it is preceded by the replication of chromosomes. This single replication is followed by consecutive cell divisions, meiosis 1 and meiosis 2. During interphase, the homologous pair of chromosomes in the parent cell replicates, resulting in a diploid cell with replicated chromosomes. Each chromosome contains 2 sister chromatids. Meiosis 1, homologous chromosomes separate. 2 haploid cells, each with replicated chromosomes, results. Meiosis 2, sister chromatids separate. 4 haploid cells with unreplicated chromosomes are the final product. Meiosis 2 is analogous to mitosis. During prophase 1, chromosomes begin to condense as homologs loosely pair along their lengths. Centrosomes movement, spindle formation, and the breakdown of the nuclear envelope occurs as in mitosis. Crossing over is completed during prophase 1 while homologs are in synapsis. Synapsis ends and the chromosomes in each pair move slightly apart. Each homologous pair has 1 or more chiasmata. In late prophase 1, microtubules attached to kinetochores at the centromeres of the 2 homologs. The homologous pairs move towards the metaphase plate. During metaphase 1, the pair of homologous chromosomes are arranged on the metaphase plate. Both chromatids of one homolog are attached to kinetochore microtubules from one pole. Those of other homolog are attached to the opposite pole. Anaphase 1 involves the separation of homologs. The homologs move towards opposite poles guided by the spindle apparatus. The chromatids move as a unit towards the same pole due to sister chromatid cohesion. At the beginning of telophase 1, each half of the cell has a complete haploid set of replicated chromosomes. Each chromosome is composed of 2 sister chromatids. Cytokinesis occurs with telophase 1, forming 2 haploid daughter cells. No replication occurs between meiosis 1 and 2. Prophase 2 involves the formation of spindle apparatus. In late prophase 2, the chromosomes move toward the metaphase 2 plate. During metaphase 2, the chromosomes are positioned on the metaphase plate. The 2 sister chromatids of each chromosome are not genetically identical due to crossing over. The kinetochores of the sister chromatids are attached to microtubules extending from opposite poles. During anaphase 2, the chromatids separate and move towards opposite poles as individual chromosomes. Finally, at telophase 2, chromosomes condense and cytokinesis occurs. The meiotic division of one parent cell produces 4 daughter cells.
Mitosis, DNA replication occurs during interphase before mitosis begins. In meiosis, it occurs during interphase before the start of meiosis 1. Mitosis involves 1 cell division and meiosis involves 2 cell divisions. Mitosis, meiosis 1, and meiosis 2 each contain prophase, metaphase, anaphase, telophase, and cytokinesis. In mitosis, the synapsis of homologous chromosomes does not occur. Synapsis occurs during prophase 1 of meiosis along with crossing over between non-sister chromatids. The end result of mitosis is 2 diploid daughter cells that are genetically identical to the parent cell. The end result of meiosis is 4 haploid daughter cells, each containing half as many chromosomes as the parent cell. These daughter cells are genetically different from the parent cell and from each other. Mitosis is responsible for the reproduction of somatic cells, allowing multicellular adults to arise from zygotes. It produces cells for growth and repair. Meiosis is responsible for the production of gametes or sex cells and allows for genetic variation. Without these processes, our growth and development, as well as the reproduction of offspring, would not be possible. This has been Danielle and Lauren. We hope you have enjoyed our presentation.